Good morning students. Today I am going to explain about transgenic plant parts 2. This is the last video of this fourth lesson, that is principle and process of biotechnology. So this video starts with the green fluorescent protein. This is very important question because this year last public question is this green fluorescent protein. They have asked you in the 1 mark question. Okay, what is this green fluorescent protein? It is nothing but it's just a protein. It consisting of 238 amino acids with a weight of 26.9 kilodalton. Okay, so this protein have the tendency to exhibit the bright green fluorescence color. It is going to exhibit some fluorescence color, that is in the bright green color. Okay, when it is exposed to a ultraviolet range, that is 395 nanometer. Okay, this is a protein. This protein have the tendency to give or exhibit their dark green or the bright green fluorescent when it is exposed to 395 nanometer. Okay, so because of this ability to form a internal chromophore without requiring any accessory cofactors. Okay, so it have that fluorescent exhibiting capacity. This capacity to this plant, it does not need any accessory cofactors like NADP, NADH2, ATP. This kind of cofactors does not need for this fluorescence, and also no need of gene products or the enzymes or the substrate or anything or other than the molecular oxygen. Okay, why this GFB gene, that is this green fluorescent protein is used precursively? That means it has been used in the modified formats to make the biosensors. What is called biosensors? If we are using a biofermentor, that is in the I have explained you in the video class itself, that is, so in that fermentation process is done. If there is the temperature is increased or the temperature is reduced, that will be indicated by these kind of the GFP proteins or the green fluorescent protein called biosensors. This is just protocol how they are making this green fluorescent protein, that is it is actually isolated from the Aquaria victoria. It is a jellyfish. From this jellyfish only we are taking this green fluorescent protein gene, and gene splicing technique, by using of this gene splicing technique we are just altering or entering this GFP gene into our our particular DNA. Introduced into the Arabidopsis thaliana plant. It is a plant. In this plant we are introducing this gene, that is GFP gene, into the Arabis, that is Aquaria victoria. Is jellyfish gene as a GFP gene. It is a desired DNA which is incorporated in the Arabidopsis thaliana. Okay, and this GFP is expressed in the Arabidopsis thaliana. This is jellyfish, and the DNA is taken and it is inserted into the Arabistopa, that is thaliana. Okay, what is called biofarming, you should know. So biofarming is nothing but it is a pharmaceutical substances used for human beings. Okay, so like example, if any plant having any plant or the animal sources or the aquatic animals like jellyfishes, it consisting of some pharmaceutical substance. What is called pharmaceutical substances? Simply in one word we can say it is a medicine. Okay, it has as a medicinal capacity to cure some diseases or infections, so that it is called pharmaceutical substances. So that pharmaceutical substances we are taking from the living organisms, so that it is called biofarming. Okay, this is also called molecular farming or the farming. Okay, because that particular plant or the animal sources which is having this farming pharmaceutical substances present in the organisms, that particular organism have the tendency to produce that pharmaceutical substances with the help of their particular, according to their respective gene. That gene is we are taking and incorporating in our desired host. Okay, so that it is called biofarming. These plants are different from the medicinal plants, and which are naturally available. Medicinal plant means that is different, and this is different, totally different. Okay, so and it is also used in the bioreactors, gaining more significance in the biotechnology. Many pharmaceutical substances can be produced using a transgenic plants. By using of our transgenic plants only, we are taking the pharmaceutical substances. Many type of pharmaceutical plants or pharmaceutical substances producing gene of one particular organism is taking and introduced into the another particular organism like plants. So through these transgenic plants only we are getting the many pharmaceutical substances. Example, golden rice. Okay, golden rice I'll explain you later. Bioremediation. What is called bioremediation? That is, you know that our environment is get polluted because of this noise pollution and environmental pollution, that is air pollution. Everything takes place. Okay, that pollution is removed by using of microorganism or plants to clean up our environment. How will you remove the air, or how will you stop the pollution or clean the pollution of environment by using a microorganisms or the plants is called the bioremediation. Okay, so they used to treat the waste, including wastewater, industrial waste and solid waste. Okay, and also this microorganism and plants also used in the wastewater, industrial waste, solid waste, to clean up the industrial waste, to clean up the wastewater, to clean up the solid waste. All those things we are applying. Okay, so in this bioremediation process, they just remove the oil from this material and petrochemical residues and pesticides and heavy metals from the soil or the grounds. So this is the main mechanism of our microorganism or plants which we are applied for the bioremediation process. First it removes the oil, next it removes any petrochemical residues or the compounds and pesticides and heavy metals from the soil or the groundwater. Okay, so it is a very less expensive and more sustainable than the other physical and chemical method of the remediation. And bioremediation process is a cheaper and eco-friendly. Bioremediation process abdina, it is very cheaper and it is eco-friendly. Okay, so and its deal with the lower concentration container is more effectively. Next, the continuation of the bioremediation, it is indigenous microbial population as indicator species for the bioremediation process, and it is addition of the adapted or the design to a microbial inoculants. Example, uses pathopna, phytoremediation. Phytoremediation, that is phyto meaning is plant. Plant remediation, we are using the plant will bring out the remediation of the environmental pollution, that is the carbon dioxide, sulfur dioxide, nitrogen dioxide, nitrogen dioxide, nitrite. These are all polluting our environment, which is removed or taken up by our plant. Mycoremediation means myco means fungi. Fungi will bring out the removes the pollution in the environment. Biobending is the process. It increases the oxygen or the air flow to accelerate the degradation of environmental pollution. Next, bioleaching is nothing but it is just remove the metal pollutions like metal pollutants like tungstens or any other mercury. This kind of the poisonous the dangerous metal pollutant will be removed from the contaminated sites like infected sites. And bioaugmentation is addition, it's a selected microbes to speed up the degradation process. Okay, it is going to degrade something, so that degradation process is speed up by the process called bioaugmentation. Next, composting is the process by which the solid waste is composed by using of microbes into a manure which act as a nutrient for the plant growth. Yes, so composting is meaning is nothing but degradation of our or composting or the degradation of the plant and animal waste so that it is useful and nutrient and enriched nutrient for the plant growth. And rhizofiltration means that is the metals or the degradation of organic compounds by a rhizosphere organisms. And rhizostimulation is a stimulation of plant growth by rhizosphere by providing a better growth condition, reduction of toxic material from the soil. Next, limitation. What is the limitation for this bioremediation is nothing but only the biodegradable contaminants can be transformed using a bioremediation process, and this is specifically made for the conditions at the contaminated sites. sites. Small scale test on the pilot scale must be performed before carrying out the procedure at the contaminated site. We have to use the once we are using this bioremediation process, we have to check with some other places. That's very important. That is one of the limitations. Then use of genetic engineering technology to create the genetic modified microorganism or the construct of the microorganism for the bioremediation process is a great potential. It's a great potential only, but it is take more expenses. Okay. Next, biofuel means algae they are using for to get the fuel. Fuel, you know that petroleum, diesel and all is a fuel for to run our vehicle. But that fuel also we can take it from the microorganisms like algae, so that it is called algae fuel. This algal fuel is also called a biofuel. The algae fuel is nothing but it is algae oil. Okay, it is an liquid fossil oil and it is having some petroleum products compounds. Okay, and it is commonly known as biofuel, and it is actually taken from the corn and sugar cane. What is it? We are taking from the corn and sugar cane, and the energy crisis and the world food crisis have initiated interest in algal culture, that is farming algae. By farming or by reproducing of algae is making the biodiesel and other biofuel using land unsustainable for agriculture. Okay, so we can use this biofuel for our agricultural work. And Botirococcus brownie is a normal used to produce the algae biofuel. Example of the algae, Botirococcus brownie is a normally we are using for the producing algae biofuel. This is just a diagrammatic representation of algae biofuel. This is a algae using sun energy. This from they are taking oil, and it is the oil extraction and protein residues there in the oil of this algae oil, and it is also used for biodiesel production. Glycerin product is there, and renewable fuel is there, and it is used for the existing carbon dioxide energy. That's all. Biological hydrogen production by algae also are important, and it is a photobiological water splitting. It's a method. Okay, you know that you heard about that is Chlamydomonas is algae, unicellular photosynthetic algae. That Chlamydomonas reinhardtii is releases the oxygen when it is deprived in sulfur, so it switches to the production of hydrogen during the photosynthesis, and the electrons are transported to the pyridoxine hydrogenase enzyme and combine them into production of hydrogen gases will be produced through this way. Okay, so this is nothing but this is your electron transport chain. Here your pubiquinin is there, and in your cytochrome is there, the phytocyanin is there. So this is your photosystem 2, photosystem 1. This is you have studied in the last year. Same thing only here. Here through this mechanism they are taking the hydrogen production through the photosynthesis process. Bioprospecting is nothing but it is a commercialization of new product obtained from the biological resources. Okay, so they are discovering and commercialization some products, new products which is obtained from the biological process. Okay, so this include the biopiracy, in which the indigenous knowledge of the nature originating with the indigenous people used by the other profit without authorization or the compensation to the indigenous people themselves. Okay, the biopiracy is nothing but manipulation of intellectual property right laws by the corporation to gain the exclusive control over the national genetic resources without giving adequate recognition or the remuneration. Example of the biopiracy include a recent patent means that's a human right. Okay, human discovered something, that human have the full right on that particular discovery. Okay, not on the government or not at all. That is called recent patent. Okay, by US patent and a trademark office to the American companies on turmeric, neem, and most notably in the basmati rice, and all three products are indigenous to the Indo-Pak subcontinent. So biopiracy of the neem is nothing but the people of the India used neem and its oil in many ways for controlling fungal and bacterial skin infections, and also it's have the properties to entire. So India have shared the knowledge of good antibacterial antifungal activity of the neem to the world. Okay, the pirating this knowledge, United States Department Agricultural and an American multinational corporation, W. Grace, in early 90s showed a patent from the European Patent Office on the method for controlling the disease on plants by aid the extracted hydropic neem oil. So the patenting of fungicidal and antibacterial property of the neem was an example of biopiracy, but the traditional knowledge of Indians was protected in the end. The biopiracy of the turmeric also having the antifungal antibacterial activity. That activity will antiseptic also we can use this for the wounds. We can use this. United States Patent and Trademark Office in the year of 1995 granted patent to method of use the turmeric and an antiseptic agent. If we have any cut or abrasions in the skin, we can apply the turmeric oil, turmeric powder to that particular areas. So this also a home remedy for the quick healing of the wounds and healing rashes and abrasions, and also the journal of the article published for the purpose of the healing rashes and Indian Medical Association in the year of 1953 remedy for mentioned proved that use of turmeric as an antiseptic not new to the world, a new innovation, but formed part of the traditional knowledge of Indians. And Basmati also in the year of on September 2, 1997, US Patent Trademark Office granted patents on Basmati rice lines. The grains Texas-based company Rice-Tech, they said that to determine the cooking properties and starch content is rice grains. Okay, and also United States to take a matter WTO as an infringement of the TRIPS agreement, which could have resulted in the main embarrassment of the US. Hence, the voluntarily and due to few decisions take the US Patent Office Rice-Tech and no choice but lost most of the claims and most importantly the right to call the rice basmati in the year 2002. The fine decision was taken. Rice-Tech dropped down 15 claims, resulting in the clearing the path of Indian basmati rice exports to the foreign countries. The Patent Office ordered the patent name to be changed to the Rice Lines 867. Applications of finally biotechnology. Biotechnology is one of the most important applied interdisciplinary scenes of the 21st century. 21st century to the it is trusted area that enables use to find the beneficial way of life. That field like agricultural, medicine and environment and commercial industries. Transgenic varieties like plants, transgenic cotton, BT cotton, rice, tomato, tobacco, cauliflower, potato, banana. So development, transgenic, pesticide resistant, stress resistant, disease resistant varieties of agricultural crops is immense outcome of the biotechnology. And human insulin and blood protein in E. coli utilized for the insulin deficiency disorder in human breakthrough the biotech industries in the medicine. So that's about this lesson. Thank you, students.